All right, let's get started. How's it going? Good, thanks. I rarely get someone ask how I'm doing. Thank you, whoever said that. Hopefully you had a great long weekend, and here we are, all set to tackle exam 1 tomorrow. Right? Yes? If not, you still got today and technically all day tomorrow if you wanted to. Anyways, we'll get to that afterwards. What we want to do today, we want to finish off our content for Unit 1 first. So there was Section 23 that we haven't touched yet. Section 23 is a brand new section that we actually created this summer. I was all pumped up to kind of dedicate a whole course to it. And then, of course, with the lockout and all our condensed scheduling, that's one that got the cut. But essentially what I want to do is that Section 23, hopefully you read through it, and what we're going to do today is just kind of map out the pros and cons for each technique when we're talking about structure versus activity. So I'll provide you that table, you can take notes, and then we'll go on from there. That will be the conclusion of the Unit 1 content. So before I get going, I just want to kind of get an announcement out. I am aware that some of you guys can't access the lecture recordings and the post-lab videos right now. Turns out if you accessed it before, so early in the semester you were clicking on the videos and whatnot, and you signed in, it should still be working. If you've never done that and the sign-in process, you have to do it, right now it's not working for whatever reason, out of my control. So what I'm going to do after this class, which wasn't on my to-do list for today, but here we are, I'll have to chase down tech to tell them that they have to get the problem solved soon, please. And in the meantime, what I'll do, I'll download all the videos and I'll post them somewhere public. And I'll just post an announcement with a link where you can click on all the in-class lectures and the post-lab videos while we're waiting for the others to fix the Panopto situation. They tell us to put all our videos there. And then right now, my students can't access their videos. Isn't that convenient? Anyways, so we'll figure that out soon. So if you can't access stuff, I am aware and I'm trying to help you as soon as I can. Watch out for the next announcement. Okay, so with that being said, we'll do the tables, then we'll quickly look through one question for Quiz 2. Quiz 2 in general was very well succeeded. If you're still not happy with your grade, that's okay. Sometimes it does take a bit longer. But overall, I think the average was 91. Standard deviation was 10, I believe, so variability, right? But overall, most of you guys had most of them correct, probably just one or two multi-selects, probably some missing selections or selected one that you shouldn't have selected, etc. So we'll look through one question. If you have other questions about other questions that I'm not going to show today, what's going to happen afterwards is that, first I'll reveal the Neuron Art Battle winners. So that's another thing we'll do. Once we're done that, that's essentially our conclusion for our class. I released a practice, a quiz with a bunch of practice questions. I think there's 23 extra practice questions in that quiz. So once we're done here today, you can feel free to stay and do the quiz here. So then you'll see what you got wrong. Then you can come see me in front and we can discuss what you got wrong out of it if you can't figure it out. Or if you have other questions about your notes, I'll be here to answer as well. So essentially, I'm hoping that the last kind of 30 minutes, if not more, will just kind of be open office hours, where you can either stay around, ask questions, do the quiz, or if you have better things to do, you can leave if you want. Sounds good? Okay, so let's get the content out of the way. So we're going to do the table for measuring brain structure. Okay, so we're in Section 23 stuff now. So when it comes to measuring brain structure, keep in mind, we're measuring structure here. We're not measuring activity. Okay, if ever I ask a question about measuring brain activity and you see these three options there, they're definitely not your selection because they don't measure activity. We're talking about the structure of the brain here. So one of them is called computed axial tomography, CAT scan. I'm sure you already all heard that somewhere. So what's the pro of doing a CAT scan? Who can help me out here? Yeah. Fast and cheap. You pretty much nailed it. So fast and relatively inexpensive compared to MRI, and we'll get to the MRI details right after. Very good for detecting like very obvious structural abnormalities. So if there's a big tumor or there's a stroke, CAT scans can detect that no problem. So that's why it's usually your first line of your first option to check something when we're talking about specific head injuries or specific disorders or disease that trigger the brain for something. So if you want to see the structure and you want to see something abnormal, you can use a CAT scan because it's cheap and most hospitals have them. Okay, it's widely available. What are the cons though? Yeah. Say that again? Yeah, it lacks detail, essentially. You got it. So provides less detailed images than MRI. Another potential con here is it involves X-ray radiation, right? The way the CAT scan gets the picture of the brain to view the structure is by X-ray. Is it good to get a lot of X-rays? No, it's not good for you, okay? One every couple of years, yeah, probably won't do any harm, but you can't do this in a constant manner and keep checking on things because it's actually harmful for your body. And it's limited for studying the soft tissue detail, right? So if you want to really look at a specific structure in detail, CAT scan is probably not your answer. CAT scan is just kind of the quick, cheap thing. You want to look for something that's apparent quickly. Stroke, tumor, etc. You'll see that, no problem. Then you can dive deeper, and maybe you want to do MRI to explore closer what structure is affected and whatnot. But especially for a stroke, right? You'll see that right away, and then you know you have to kind of do the troubleshooting from there. Okay, so that's for CAT scan. Now we're going to magnetic resonance imaging. So what are the pros of MRI? Yeah?
It has better detail than X-ray-based techniques because of the magnetic.
Yeah, you got it. So it produces high-resolution image of soft tissue. So the way the magnetic resonance imaging works is that you go inside a big magnetic scanner, right? It's a big tube, a big scanner. You go inside, and the way the magnetic pole works, I won't go into the physics of it. If you're really interested in all the details of each type of technique, in the textbook we provide a link to learning, I believe it's called, where you can watch a YouTube that explains all the physics behind kind of each technique. But essentially, yeah, it's a big magnet, and it kind of, the activity of the brain, it aligns the water molecules, and it will be different for different structures, and you'll get a very clear image from it. And it doesn't involve X-ray radiation, right? It's just a big magnet. And here you can actually see specific structural differences in brain disorders, okay? So unlike CAT scans where, yeah, you can see general images where there's general problems, but here you can dive specifically and target areas, right? So let's say you're interested in Alzheimer's and you want to see how it affects the hippocampus. You can actually take a bunch of people with Alzheimer's versus people that don't have Alzheimer's, and you scan and you look at the structure of the hippocampus. You'll probably see that the Alzheimer's group will have one that's kind of shrinked compared to the other one. Okay, so you can really pinpoint a structure, get a detailed image of it, and start quantifying its size. What about the cons? Yeah?
More expensive than...
You got it. Expensive and requires specialized equipment and trained technicians, right? We're legit talking about a big magnet that's going to come on, okay? You want trained professionals. You want to make sure that the people around it don't have any metal in them or on them, okay? That won't be a pretty scene. What movie was I watching? Is it Final Destination, the most recent one? Yeah, there's a scene where you see that happening. It's not pretty. Anyways, so you want to make sure that you're well-trained. The magnet comes on. It's going to align the water molecules in your brain. It's going to put those pretty pictures together. That's as detailed as I'm going to go into it. But essentially, yeah, you get these great images, but it's expensive. You need that big equipment. You need trained individuals. It's time-consuming compared to CAT. And it requires the person to stay very still in a little tube area. Okay, so if you're claustrophobic, it's not the ideal situation. And it takes longer than CAT. Okay, so you have to stay there in for a longer period of time. And it's very sensitive to motion, so you have to stay still. So not ideal for everyone. Okay, it does loud noises too. You have to stay still. It kind of sounds like a door wagging. Those are my sound effects for you. Okay, so pros and cons of MRI. So good images, however, expensive, time-consuming, and you're kind of limited to what you can measure, right? If you're someone like me who wants to study actions, how you're reacting to stuff in the environment, how you're reaching for objects, can you do that in a scanner? You're pretty limited, right? You can kind of move your fingers a little bit, but you can't really do gross actions. So you get limited to what you can kind of measure. Okay, so DTI, diffusion tensor imaging. So what's the difference about this one? So CAT, we're getting a just crude structure of the brain. You'll see if there's any obvious stuff happening that shouldn't be there. MRI, you get precise structural kind of details of areas you're interested in. Now DTI, why is that one different? It still uses magnetic scanners, it just uses a different program to do something with the image to trace something else. What is it? Yeah. It does track water molecules coming across connections. So not only are you getting structures, high resolution pictures of the structures, you can actually start mapping out the connections of each structure by using this technique. So it reveals the white matter tracks, right? Remember I showed you the gray matter versus the white matter? The white matter is all the axons coming down and connecting to other structures. So DTI does a very good job at mapping out the structural connections. Okay, so MRI, very good at the structures itself. DTI, you actually get to see the connection. If ever you saw like a brain picture with all the fibers and different colors coming across it, that's a DTI image, okay? So they track these water molecules coming through these connections, and how they track that, it shows you where the connections are coming from and where they go to, and then you can start mapping out, okay? If it's one area connecting to another area, you'll have all these kind of green fibers, let's say. Then another structure might be pink fibers coming across. Then you start to conceptualize how everything's connected. So very good for doing that. And it can detect white matter damage, right? So if we go back to the multiple sclerosis example I was talking about last lecture, where they have this white matter degeneration in the central nervous system, well, if you do DTI to a brain with someone with MS, it will become apparent that there's actual kind of connectivity issues. So where there's supposed to be clear white fibers coming across, you actually have like broken connections. So it allows for visualization of neural pathways, not visible in standard MRI. So what are the cons? Well, once again, you're dealing with MRI technology, so costly, less accessible. Once again, the sensitive to motion stuff. And it's very complex data analysis to reconstruct those pathways I'm talking about, okay? It's not just you sit in the scanner and it magically gives you that. There's a lot of tracing where it's going, a lot of stats being applied. Then eventually they give you a good conceptualized image of what it looks like. But a lot of effort put into it to get those pretty pictures. But very convenient for tracking those connections. Okay, so that's measuring brain structure. Okay, we're not talking about activity right now. We're just talking about measuring a structure or structures. Are we good with this? Does it supplement the material? So like I said, I don't want to cause too much panic because I don't want to throw new content at you right before an exam. So this is section 23 stuff. It should complement, right? Whatever you read, this should just be a bonus, right? It gives you a proper sheet to rely on if you get a question and you want to kind of compare it quickly. Good? All right, moving on to activity. So for the activity, you have kind of 5 main types. So if we look at the first one, positron emission tomography, so PET. So a PET scan, once again, pros and cons. And this is going to be kind of you might not see clearly here because the word letting's going a bit smaller because I'm trying to shove everything in a one snapshot. But bear with me, it should be quite visible. Okay, so pros and cons of PET. So what PET does is that it measures the metabolic activity of neurons, okay? So what usually happens is that before you go into the scanner, you're injected with what we call a tracer. So it's usually a radioactive tracer that will trace some type of byproduct of what the neurons are doing. Okay, so for example, neurons when they're active, they demand high energy, right? Neurons are always hungry, especially the active ones. So where glucose is being absorbed within the brain, where the neuron stuff is happening, where all that activity, you can trace glucose. So you can have a tracer that will trace that. So as you're sitting and doing whatever task you have to do within the scanner, they can eventually see where the activity is happening by following where the tracer is going in the brain, essentially. So it's very good to provide molecular and kind of biochemical details. So in the textbook, it gives you a picture of, it was Alzheimer's. So Alzheimer's, what happens is that there's an accumulation of what we call these amyloid plaques. So amyloid plaques are something that kind of starts to gather in the brain as you age. But when it comes to Alzheimer's, you have that happening kind of in an exaggerated way, much earlier than kind of a healthy aging brain, let's say. So you can have a tracer that will actually trace for those plaques. So what you see in the textbook is a group of people with Alzheimer's, a group that don't have Alzheimer's, and then you see the Alzheimer's groups, you have that full-blown, is it a bright red or a bright yellow or something protruding through the brain? So that's the tracer kind of illuminating where all the plaques are located in the brain. So very good for stuff like that. okay? So it measures kind of the byproduct of the brain activity by kind of tagging some kind of molecular or biochemical messenger. If you're looking at specific receptors, there are some tags that you can use, some tracers that will only tag to some receptors, so then you can start mapping out where are the receptors in the brain. That's another way you can use PET. But the cons are, it involves a radioactive tracer, right? So you can't do this every day, okay? It's not that healthy for you. Like I said with the CAT scan, once in a while, every couple of years. Not totally horrible, but you don't want to do this constantly. And it's very poor for the temporal resolution. So when I say temporal versus spatial we mean the specific location of where the activity is happening, whereas temporal, we're talking about the timing. How fast can you capture that activity? So when it comes to PET, yes, you can pinpoint some specific activity by looking at the metabolic activity you're tracing, but the timing is a bit delayed, okay? So you won't get real-time activity. You kind of have to wait a little bit, then once they do the task, eventually you'll see where the tracer's going. So there's a delay there. So it's not great for temporal resolution. And it's expensive and requires specialized equipment. Okay, so unless you have a very specific question about receptors or neurotransmitters or specific molecular byproducts of what neurons are doing, you would use PET. Aside from that, it's pretty rare you would use it. So now going to the more common one, so functional MRI. So we know what an MRI is now. Now we're putting the functional part to it. So not only going in the MRI where you can get the picture of the structure, but you can also use a technique where it will actually map out the activity based on where the fresh oxygenated blood is going in the brain. Okay, we call that the bold signal. So where is the bold signal going? So once again, when neurons are active and they're working, they need that fresh oxygen coming from the blood. So where the fresh oxygenated blood is traveling in the brain, that's essentially giving you the byproduct of where the activity is happening. So MRI, once again, non-invasive, right? So it's not harmful like PET and CAT, no radiation involved. There's high spatial resolution for locating activity, okay? Very good at pinpointing the exact location of where the activity is happening. Okay, based on that bold signal, you can really trace and compare your structures, right? So you might want to study the motor cortex for whatever reason. You might have a task where you're showing, I don't know, a video of someone doing an action versus a video of not doing an action. You want to see how that resonates in the brain. So you take the motor cortex, you kind of measure it for the bold signal, and you start comparing your differences. Is the motor cortex more active when watching the actions versus the non-actions? Most likely, yes. So then they put those pretty kind of images, right? So the pretty brain pictures all lit up with pretty full, all types of colors. That's just based on where the bold signal is, right? It's not you go in the scanner and it gives you that picture. You have to provide the stats of where the bold signal is, and then where it is the most, you put a specific color. So if it's high activity, you might put a bright red, low activity, maybe yellow, whatever it may be, and then you kind of map out where the activity is happening in the brain, depending on what type of task you're looking at. So it's kind of become the gold standard of studying brain function, okay? There was a big boom of using fMRI research in the late 90s, early 2000, where it opened the door to really pinpoint where stuff is happening in the brain. Some of us, we've got carried away, though, to be honest, because at the end of the day, it's only a correlation. We're not actually measuring brain activity. It's an indirect measure of brain activity, right? You're relying on the logic that it's using bold signals, I'm going to track that. But just because one part's active doesn't mean that's the only part, right? It could be connected with 1,000 other parts, could be inhibited from other parts. So there's a lot of other questions aside from just showing where the activity is happening. But it's definitely a good start to start understanding the brain. So there we go with the cons. I kind of foreshadowed that. It's an indirect measure of neural activity. So you're actually not measuring any electrical activity. You're just going by that bold signal. Expensive, requires a lab or hospital setting. Once again, you need the MRI scanner. So you're dealing with a big magnet again. You need some specialized trained people. Sensitive to motion, limited to a still task, okay? So same issue with the MRI. If you want to pinpoint activity and you want to do actions, then you're kind of limited with what you can study. You have to stay still. And not great for temporal resolution, okay? You won't get real-time images. There's always a delayed, okay? So when the activity of the neuron is going, the bold signal kind of happens afterwards, so there's a delay period there. So by the time you catch that signal going there, the activity already happened, essentially. So it's not measuring real-time activity, it's not directly measuring activity, but it's giving you a precise kind of location of where the activity took place, essentially. Okay, so good spatial, but poor temporal, especially compared to EEG and MEG, which we'll get to at the end here. All right, now functional near-infrared spectroscopy, so FNIRS. So FNIRS is a very kind of convenient tool to have as a neuroscientist because it uses the same concept as MRI, but instead of relying on a huge scanner, you're actually doing it with just a light source. So you have a cap that you put on. Some FNIRS will be a full cap, kind of like an EEG cap, but instead of the electrical sensors, it will actually be just light sources, kind of all across the cap. Or if you're only looking at a specific strip of cortex, it might just target that specific strip. So if you're looking at frontal stuff, it might just be like a headband strip. Could be all types of things, but essentially what it does, the light sources that are penetrating the skull, you can kind of get a measurement based on how they're reflecting back. So if it's an oxygenated bold signal, it's going to reflect differently than a non-oxygenated bold signal. So once again, you can track activity. But you're not relying on a magnet, and you have a cap on. You're relying on those light sources. So obviously, based on that explanation, it's portable and motion tolerant, okay? You're not stuck in a scanner. You can actually do stuff with it. In your textbook, they actually show a FNIR device where they're doing a virtual skiing task. So they're doing some skiing movements, kind of moving their head and whatnot, and that's totally fine. You're still getting a good signal. Obviously non-invasive, okay? So there's no kind of radiation or anything, and you're not stuck in a scanner, right? So it's a lot more suitable for children and infants, okay? You can put the cap on a child, no problem. They can do stuff without being too bothered. Versus try putting a child in a scanner. That would be difficult. Once again, this has very good temporal resolution, okay? So unlike MRI, where we're all talking about the spatial precision, but not good temporal, this one actually has very good temporal, okay? Whenever the activity is happening, it's kind of catching it almost in real time. So what are the issues with this one? Well, you have limited spatial resolution because the problem with this is that the lights can only go deep enough. So you can't measure deep brain structures. If you wanted to answer something about the activity of the amygdala or the hippocampus or something like that, FNIRS won't be your option because it can't go that deep. So essentially you're limited to stuff on the surface of the cortex. Okay, so limited on the structures that are on the surface. If we're talking about deep brain structures, you won't be able to measure that activity. And it's less detailed than fMRI, okay? When we're talking about kind of the spatial resolution, yes, you can get the general kind of area where stuff is happening, but it's not as precise as fMRI. Okay, are we good so far? Yeah?
Sorry, can you explain the temporal situation a little more? I don't get how it can be delayed. I don't understand.
Because it's a light source, it can capture the bold signal a lot more quickly than the way the magnetics are pulling it from the fMRI. So there's a delay in the fMRI to get the activity. So let's say you tell me, picture yourself, I don't know, riding a bike or something, and you're looking at the structure of the motor cortex or something. So while I'm doing that in the scanner, it's going to take a while until the activity actually appears on the program you're using because of the delay of the magnets and how it works with the bold signal. Whereas the light with the bold signal can get it immediately. Not immediately, but close. Yeah. EEG and MEG are even better for that. We'll get there. But yeah, essentially that's how it works. And like I said, you don't have to go deep into this stuff. It's a good question and you're curious, so that's why I like to answer it. But I'm going to keep it very straightforward. Like I said, I don't want to throw brand new content at you guys, as long as you know the pros and the cons. So if I show you a question and I give you a situation of what type of technique should they use to measure whatever we want to measure, you know kind of where to go, right? Okay, EEG. So EEG directly measures neuronal activity, okay? All the other ones are not a direct measure of actual electricity happening in the brain. EEG does that, okay? So we're not talking about specific action potentials. You could do that with a needle kind of EEG, but we won't get there. Essentially, the EEG, what it does, it just tracks where the flow of electrical activity is happening. Because what happens when you do something, the activity associated with that location or that structure will kind of synchronize together, and you get a good kind of signal coming from that area. So the EEG will pick up those electrical signals. So once again, excellent temporal resolution because you're actually directly measuring electrical activity, so there's no real delay, unlike the bold stuff. Cheap and portable and non-invasive, okay? And it's useful for diagnosing stuff like epilepsy, sleep disorders, and brain injuries, okay? So the thing with EEG, you can measure specific brain waves, and you'll get more into that in Unit 2 when you talk about sleep stages. You'll see the specific brain waves associated with each stage. So that's why it's good for sleep disorder stuff, okay? Whenever we're talking about sleep disorder, they'll probably get you to come into the hospital or the lab, whatever the setting is, to stay overnight at least once, where they'll put the EEG cap on you and actually see how you're cycling through the phases of sleep. And depending on where stuff goes wrong, then they can maybe diagnose you more specifically. So EEG is great for that stuff. Is that a question in the back? No, sorry. Now the cons. So although it's very good for temporal, you get the trend here. When one's good with one, it's usually the opposite for the con. So very good with temporal stuff, okay? Gets the timing good, but the spatial resolution, not that great. And once again, you have the same problem as FNIRS. You're only limited to stuff that's happening on the surface, okay? So with an EEG kind of typical setup on your head, you wouldn't be able to measure deep brain structures again. Since we're relying on actual electrical signals, it can actually be very noisy, okay? So it is a bit distorted by movements and whatnot. So unlike FNIRS that is a bit more resistant to movement, EEG, yes, it's a bit more portable and you can move a little bit, but if the cap starts kind of moving around, your signal's gonna be noisy and you're gonna lose what you wanna look at. So you are a bit limited with what you can do with it. And if you read section 23, or any section of our textbook as you continue on, you'll often notice that we have like dig deeper boxes. And these are boxes where we'll explore a topic a bit more precisely and we'll add stuff to it. And for our dig deeper box, we actually go deeper for this next point, where it's not compatible with all types of hair, okay? So this led to exclusion of a lot of participants, which shouldn't be excluded. If we're talking about the science, we want it to be the science for everyone, not just a specific hair type. So that was a problem for the first wave of EEG research, where if your hair did not fit with the EEG cap, you would just be excluded. Whereas now we have better solutions at least, okay? We got researchers with creative solutions on how to put clips to make sure the electrodes can still read through the hair and whatnot. So if you haven't read the Dig Deeper box, please go read that because it really conceptualizes how to make science better with little adjustments. But essentially that's one of the downsides of EEG, okay? Depending on your hair type, there might be a lot of adjustments in order to get the signal through. All right, last one. Then we're done for the content of Unit 1. So MEG, the MEG. So the MEG is very good, excellent at temporal resolution, and better spatial resolution than EEG. So you're kind of getting the best of both worlds with the MEG. So if you haven't read the section yet, essentially the MEG, what it looks like is you're sitting down and there's essentially like a bowl coming on top of your head, and that's where all the sensors are. And once again, I won't get into the physics, but the thing about the MEG is that it actually captures the magnetic fields that are kind of happening by the activities of your neurons. Because keep in mind, neurons, when they're active, there's ions flying in, ions flying out. Turns out that creates a magnetic field that you can actually capture with specific sensors, and that's essentially what the MEG does. Just a bunch of little sensors within that kind of head frame that they put on top of you, and it measures those magnetic kind of measurements. So very technical, but very cool. And because of that technique, you do get good temporal resolution and pretty good spatial resolution. Less distortion because you don't have a cap, right? So as long as you're sitting down still and you have that on top of you, there won't be much distortions. And the thing about this is that it's very good at tracking communication between areas. Okay, so DTI, we talked about looking at the tracks. Now we're looking at where the activity is coming from, okay? Who is using those tracks as you're measuring what structures? Okay, so very good at using that for functional connectivity. Okay, like I said, it's not only important to see what activity is happening in a specific area, but where is it bringing that activity, or where did that activity come from? How is it all connected? So you really get to explore networks, essentially. So not just structures, but networks of connections, how it's happening in the brain in essentially almost in real time if you have the right program going on, right? So you get them to do the task, you get the measurements, and you see how all the related brain areas communicate with each other. So very good at measuring that functional connectivity. Now the cons, you can probably guess this one, extremely expensive and requires a shielded room. You actually need liquid helium cooling. So the sensors I was talking about, I guess they're kind of using that to capture that magnetic resonance happening from those neurons' activity. So you need all those technical crazy gadgets that are very expensive and very kind of cumbersome to maintain. We actually had a Meg at the IWK that a lot of my colleagues were using before. And turns out they decided not they're not funding it anymore, so it's not working anymore. So before I like to kind of compliment and be like, yeah, we have one here, it's a pretty rare device, but we do have one in Halifax. But apparently it's not functioning anymore. So limited availability, right? So you need the specific room, the specific trained clinicians, trained researchers that know how to run it. And once again, you do have to require participants being still. Okay? You don't have the issue of the cat moving around, but you have the issue that you can't really move around your head. You have to stay still. And those are your techniques for measuring brain activity. So you see some are good for some things, some are better for others. Depending on what the situation is, you want to use one versus another one. Okay? So I'll pull in probably one question at least where I'll give you a situation and I'll ask you which technique would be better to kind of get an answer for that situation. So maybe it's someone who's interested in how you perceive the unified language that wants to look at the connections between the Broca's and the Wernicke's area as they're doing an activity of a specific task. So we're talking about connectivity between two areas. You know that the Meg would probably be the answer here. Okay, stuff like that. Any questions? Okay, so that's it for content of Unit 1. Now let's look at our Quiz 2 review. If you don't have all the notes, don't worry. I will eventually get a video out. I know it probably won't work right now, but eventually we'll have a video out for you, so you can pause that, take the notes out. Or you can come see me after if you're missing something and recopy it. I'll stick around once we're done. So let's go to Quiz 2. So like I said, Quiz 2, for the most part, very well succeeded. This is the question that I'll go over with, because most of the questions had a very high success rate. This one did as well, but it was less than most of the others. So we'll review this one. So motor neurons and interneurons have similar blank, but different blank. Okay, so if we start with E, so similar action potentials, yeah, that kind of makes sense. We know action potentials are always the same thing. Different neurotransmitters, yeah, it does make sense. But guess what? Could be the same ones too. And I actually never covered that in class. So action potentials and neurotransmitters here are a distractor because they don't really fit in with the question we're answering. So E, that's not correct. D, even if it was correct, the action potential part doesn't make sense because it's not different. It would be similar. Then we already went through the neurotransmitter, why it doesn't fit here. Now we're with the targets and the inputs. Okay, so motor neurons and interneurons have similar but different. So if we think about it, interneurons, their inputs comes from what? A what? Yeah, sensory neuron, right? So another neuron. What about a motor neuron? Where does its input come from? An interneuron? So another neuron. Ooh, we have something similar here. Now let's look at the targets. Where does the interneuron kind of send its signal? To another neuron, to the motor neuron. Okay, now let's look at the motor neuron. Where does it send its signal once it's done? Muscle fibers. Oh, that's different. So there you go. So motor neurons and interneurons have similar inputs, but different targets. Okay, so that's what we meant by this question. I know the wording here maybe tripped up a few of you. I'll make sure stuff is a bit more clear for the exam so we avoid these ambiguous kind of things, but you get the clear answer once you break it down properly. Is that clear? Okay. Reminder, once again, you have an exam tomorrow. So what's going to happen, the link will be available from 8 a.m. to 8 p.m. So as soon as 8 a.m. hits, you should have access to hit the link. Once you click the link, you'll have a description. Once you're ready to go, you're going to click start. Once you start the exam, the 80-minute timer will start to go off, okay? So if you have accommodations approved, you can go past the time's up. So once it hits 0, it won't close on you, essentially. So use your approved time, and then once you're done, you click submit, okay? And then you're done. The questions were submitted. So, exam consists of 36 questions, right? So you'll have 24 multiple choice questions, and these are worth 0.5. So 0.5, and 12 multi-select questions, which are worth 1. Okay, so that's where you get your 36 questions. Combine all of those together, it will give you a score of 24. Yeah?
Are there anywhere we can find practice questions?
Oh yeah, there's some that have been released. I'm getting there. Yep. And you'll have a chance to do them right here because I'm almost done. So yeah, there are practice questions out. That's kind of my last slide though. Okay, exam 1, you know that it covers unit 1, lessons 2 to 5. It includes all the assigned readings from chapter 1, 2, and 3, all the associated tricky topic videos, and the class content we covered. I already showed you this, so you can know what to expect. It will be 4 to 6 questions for each kind of bullet point here. So these are all kind of our main lectures, our main tricky topic videos, main textbook sections. Okay, so there should be no surprise there. And here are your learning objectives, right? So we don't hide anything in this course. We tell you exactly what you're going to be tested on. So for each lesson, this is on Brightspace, right? You click on a lesson, you go to content, you'll see this at the end. So each lesson has specific objectives. So we don't put those there just for fun. It's to kind of give you a foreshadowing of what you will be tested on. So what I would do if I was studying and revising or reorganizing all my notes, I would go through each lesson and go through each objective and kind of picture yourself, could I come to class and come here and describe all the subdisciplines of psych and neuro pretty confidently? Could I come up with a table without using my notes and kind of dish out the main ones pretty easily? If you can picture yourself doing that in your head and you're doing pretty good, it probably means you really understand the material. If you don't go very far with that mental activity, it probably means you don't understand it quite well. So even if you have pretty notes, it means you still haven't got the concept right. So go back, fresh up, try to reread your notes, rewatch the video if there is one, rewatch the lecture. If you can access it, which you will, I'll give you an option some way or another. But yeah, go ahead. No, so what's going to happen, you're going to submit, and I will probably release the grade the next morning. But you'll only get the grade. You'll be able to re-access and go see your exam probably the Monday after. Once I post the exam link, so at 8 a.m. tomorrow, I'll give you a bit more detail about when stuff will be available. So you won't get your grade right away, but you'll get it the next morning probably. Unless there was a major issue and I have to troubleshoot stuff. There might be a delay, but you should get it within the next day. Yeah. Yes?
Are you able to maybe see what we got wrong on our quizzes so we know what to focus on more?
You can. So do you know how to go back to your quiz?
I guess I can't access my wrong answers.
Yeah, can you see your answers though? Can you see what you got wrong? I'll show you. Come see me after. There's a way you can go and see what you got wrong. I don't show you the answer, but you can see what question you got wrong. Then based on what you got wrong, you might be able to figure out the answer, and if not, you can consult me. Yeah. Okay, so I would go through each step and make sure, do I really know this or not? Could I come up with a multiple choice question? Could I create a multi-select question? And if you're having fun doing this for whatever objective you're looking at, it means you probably really understand it. If you're trying to do that and you're all mixed up and you have to rely on your notes, you probably want to refresh up a little bit. It means that you don't truly understand it yet. Okay, so that's one tip. I know it's the day before, so I can't say so much that will save you at this point, but that's a little tip that you might want to do as you're reviewing your final kind of notes. So lesson 2, lesson 3, I cut out a few things, right? So inferential statistics, I kind of showed you how you can do it with the wave activity, but I was clear, right? I'm not going to show you a p-value. I'm not going to make you do a t-test, nothing like that. Focus on the average, what's a mean, what's a median mode, all the central tendency stuff, that's fair game. Know what variability means with a standard deviation score, but that's about it for the stat stuff. I won't go deeper than that, okay? So don't worry about the inferential statistics. Ethics, you read it, that's great. I won't test you on it. Moving on, so lesson 4, that's all the neuron stuff, right? Structure of the neuron, how the synaptic transmission happens, Keep in mind we have distinct processes for kind of each step. Go through them again, make sure everything's clear. If something's not clear, rewatch the tricky topic, rewatch the lecture, go reread the textbook, try to make it clearer, consult other sources, etc. Now, lesson 5, that's what we just ended, right? So last lecture, we were going through the brain structures. First, we divided the nervous system, right? Then we went through the hindbrain, the midbrain, and the forebrain. Went through the anatomical and functional divisions. Keep in mind that's different, right? If I'm asking about anatomical stuff, don't go answering with the functional stuff, and vice versa. So make sure that's clear. And then all the extra kind of areas that we went through, and this is what we did today, those 3 last points, okay? So just very simple, as long as you know the pros and cons of each one, you know which ones measure structure, you know which ones measure activity. So if we're talking about measuring a structure, I will give you a situation, you know which one to pick. If we're talking about measuring activity, give you a situation, you'll know which one to pick. Okay? So we'll keep those ones pretty straightforward because I knew it was last-minute content. Good? So you all have access to this, right? Every end of lesson, you see these objectives. Okay. Neuron art battle. So thank you for all the submissions. Okay? Very good submissions. Very hard to pick winners. That's why there's 3 winners. So very cool stuff, okay? So what I ended up doing is that I picked a best textbook style, best artistic style, and best creative style. So what I mean by textbook style is that you have to clearly know that it's neurons. Okay, it's clearly neurons. All the connections are properly labeled. Most of you guys did this style, okay? And there was many good submissions. So this ended up being an aesthetic kind of contest at the end of which one has the most kind of tasteful, artsy version of that. Now, artistic style, you're not committed to drawing neurons, but it should be clear that there's kind of 3 connections and that you're properly labeling stuff. But if you want to put an artistic flair to it, that's fine. And then the creative style was kind of an open category where you could be very, very creative. Okay, so those are the 3 winners. If you're leaving, please be quiet. Thank you. So once I announce the winners, I will eventually contact you, and I will ask you where do you want your gift card from. As long as I can send it by email, you'll get a $25 gift card. So I like to reward students who participate. I wish I could reward everyone, to be honest. But anyways, it's a competition. Only a select few can win. So with that being said, before I reveal them, if I reveal some and you're like, well, wait a minute, mine was way better than this, you might have been disqualified. A lot of you guys submitted stuff that did not go with the instructions, okay? A lot of you just copied the video of the McMaw artist, okay, that was on Brightspace, where she was doing just a simple post-presynaptic connection with the beautiful McMaw art. Some of you copied that. Some of you had neuron A, neuron B, neuron C connected in one line. That's not what we're asking for. So we were asking for that neuron A receiving a connection from neuron B and a connection from neuron C, okay? So neuron A was the postsynaptic, and the other 2 were presynaptic. If that wasn't right, you were disqualified. And some were really, really good. You would have probably won if you had the good connection. But unfortunately, have to go by the instructions. Okay? So let's get into it. So best textbook style. Let's get a drum roll. So best textbook style was, congratulations, Sarah L. Okay, so like I said, textbook style. So textbook style, it has to be clear that it's neurons, which is obvious. Neurons actually kind of look like that. Look at the soma. You see that big chunk of energy stuff happening, right? So that's pretty cool. Everything's properly labeled. The aesthetic background, everything kind of was just a bit one level up the other ones. Don't get me wrong, I had a lot of good submission for textbook style, okay? So a lot of you guys had a good diagram of neurons, all well connected, all well labeled. So good job if you were in that batch. But congrats, Sarah. Wonderful stuff. All right, best artistic style. Drum roll. Congratulations, Mila. So look at this stuff. That's pretty cool. So that's what I mean by artistic. You can tell it's neurons, but there's an artistic twist to it that makes it that much more interesting. Look at the myelin wrapped up. That's pretty cool. So obviously flowers, kind of, you see kind of the stuff coming out, which kind of mimics neurotransmission. That's pretty cool. And it's all properly labeled. It's all properly connected. So great work. That's very cool stuff. Okay, now for creative style. And I have honorable mentions at the end. Honorable mentions, unfortunately, you don't get a prize, but you were close to winning. So best creative style, let's get a drum roll. Congratulations, Sarah, another Sarah, Sarah H. Okay, so look at this. It's like a comic book strip. Okay, neuron, you see the addition, Psych 1011, comics, October 13th. The details are so cool here. So the neurons are obviously beautifully drawn, all properly connected, all properly labeled, and it's the extra details, right? The extra kind of showing you what the ions are doing here, which ones are coming in, which ones are coming out, at which point. And then also the neurotransmission. How is that happening? What's coming out? All that good stuff. So there's a story to it in a comic book fashion. So there was a lot of good creative kind of submissions, but this one kind of went a step above with the extra information that was incorporated. So that's why it won. So good job, Sarah. That was excellent. So now for the honorable mentions. So this one was pretty much the runner-up for the textbook style. Okay, so very clear, very clean, very well formatted. Okay, so good job, Claire. And this one was runner-up for artistic. This is very cool. Kind of in the henna style of things. I really like this one. It's really a different approach very unique. You don't get two submissions like that, essentially. So good job. Almost got the win. And then the creative ones. These ones are pretty cool. Okay, so these are the honorable mentions for the creative ones. So this one, it's not even a drawing. It's candy. Okay, jelly beads, maybe licorice or something. The soma, another sugary blob of something. Pretty cool stuff. You get very creative. And with that being said, the connections are all good. This one, Spider-Man throwing some webs, making some connections. Why not? It's all correct. Good job. And this one was kind of like a graffiti style, which I kind of like. I can picture myself driving, and there's like a bridge somewhere with that on the side. That would be pretty cool. So anyways, good job, guys. Like I said, if you submitted a good drawing and you did not win or was not mentioned, it's not because it wasn't good. Most of them were very good. And who cares if you didn't win? That wasn't the point, right? The point was actually doing the activity. So you're embodying the cognitive learning, right? You're actually going into the material, actually making those connections. So if you like doing that, quiet please. Quiet for the people staying. So if you like doing this, I highly recommend you keep doing this for Unit 2. So Unit 2, you'll get like the visual system that you can draw up. So even if Dr. Duffy doesn't give you a contest or anything, it doesn't mean you can't incorporate what you like for studying into your own style, right? So if you like these summary tables, keep doing them. If you like these drawings, keep doing them. So hopefully I provided you some extra tools that will actually help you at studying. So this is it for Unit 1, right? So thank you guys, okay? Thank everyone. You guys were a very good group. I know we were on a tight schedule and we were battling with time, but I was still able to incorporate my active learning style whenever I could. And you guys were very good at participating. We had some good discussions, some very curious questions, which opens the discussion and kind of solidifies the material a lot better. So without you guys, my style of teaching doesn't work. So thank you very much for responding. Even if you were here and you did not participate, hopefully seeing the participation actually helped you with gaining a better appreciation of the material. So anyways, thank you guys very much. And here is the last bit of, yes, there are extra practice questions. So on Brightspace, there's a quiz with 23 questions. They're just extra practice questions, right? So feel free to go through that to help with your studying. You have unlimited attempts. So if you go through it and you get a couple wrong, try it again and try to get 100 essentially. It's unlimited attempts. That's the way to do it. Then you'll know that you have all that nailed down. Great. Thank you very much. Take care and best of luck. Oh, by the way, Dr. Duffy, Unit 2, it starts on Thursday. I know sometimes you guys have an exam hangover. You want to come to class. He's going to dissect an eyeball. You want to see that. It's better to see it live. All right, take care. Best of luck. Yes?